Hi everyone, Doc Ron Bio here again to discuss an important aspect of the mitotic cell cycle. That is the genetic material, the DNA, throughout the process. The DNA is changing throughout the process, and understanding what is happening to it in each phase is an important step in fully understanding what this process is all about, and it is an important biological process. So let's get started. I want you to recall first that DNA is the cell's genetic material. It encodes the blueprint for the cell, how to use this genetic material during gene expression to make proteins. Recall too that proteins do many things in the cell, including acting as enzymes. They can participate in transport, they can be structural in nature, and really the list goes on and on there. But it stands to reason that the molecules of DNA that we're talking about here are very important to cell function and ultimately to cell survival. This fact is supported by the fact that eukaryotic cells have evolved the nucleus to protect their DNA. So there's an interesting dynamic here where for most of the life of a cell, the genetic material is encapsulated in this nucleus, and for a brief window of time, when we separate duplicated genetic material on the way to becoming two daughter cells, this nuclear envelope, this protective phospholipid bilayer goes away temporarily so that we can separate that genetic material. So it's a really interesting phenomenon. The DNA, as this process is happening, it is constantly changing. At some points in the cycle, it is duplicated into what we call sister chromatids. At some parts in the cycle, it is loose and relaxed. At other points in the cycle, it is condensed or compacted. Okay, so we're going to describe basically how that changes throughout the process here. So DNA as a physical structure is a double helix. So we can see that here in this picture, the classic DNA double helix. If you were to take this double helix and stretch it out, flatten it out, you'd essentially see a ladder. Okay, so your DNA is essentially a ladder with complementary bases. So that is to say, if this strand over here has the following sequence, the other strand would have a complementary sequence, the details of which are on a different video. I don't want to focus on that. But essentially, when you look at what a chromosome is, it is made up of DNA. Okay, so we have that double helix here, basically making up this loose pair of sister chromatids here. So basically, when a chromosome condenses, it is DNA that is doing the condensing. And if you want an advanced sort of explanation of things, our genetic material is essentially in a chromatin state. So it is our DNA wrapped around protein spools here. So if you have your protein here, protein here, protein here, and we'll get into the details of that in a minute, the genetic material can be wound around those protein spools. Okay, so that's called chromatin. It serves as a space-saving mechanism for the cell, as you have to pack all of that genetic material into each cell's nucleus. Okay, now another way of describing the genetic material is as either loose or coiled. So we're going to see here in a moment the points in which the genetic material is loose, and then that material compacts itself into what are often described as chromosomes, these classic X-shaped patterns or butterflies, however your teacher is drawing them or however your textbook draws them. I'm going to make some distinctions between these various states throughout specifically the mitotic cell cycle. Okay, so this next picture here is a detailed representation of DNA and how it makes up these chromosomes. Again, DNA is composed of two strands of complementary bases. You can see the ladder here going into the double helix here. And to add some vocabulary here, the proteins that our DNA is spiraled around are called histones. So essentially this complex here is a chromatin complex made up of DNA and proteins. Now, there are several layers of folding on the way to becoming a condensed chromosome or a condensed pair of sister chromatids. So you can see here the chromatin spirals up, and then even still those spirals spiral up to make this classic X-shaped chromosome or pair of sister chromatids that all the textbooks refer to when we're talking about the mitotic cell cycle. Let's advance the slide and take a look at the genetic material throughout the process of the mitotic cell cycle. Going left to right is the steps of the entire mitotic cell cycle. We start off at G1 here. That's the first step of interphase. So again, these three parts right here would be interphase. And basically with G1, you're going to start off with a non-replicated loose chromosome. I should note that all these are in the nucleus. I'm not drawing the nucleus here, but all this genetic material is in the nucleus during interphase. So you'll have a single loose chromosome. As we proceed to S phase, the next step in interphase, you're going to duplicate your genetic material. So now I teach my students to, instead of calling this a chromosome, we call them a pair of sister chromatids. So you have a black and a red sister chromatid. Ultimately, they will become individual chromosomes. So I say that sister chromatid step because it shows us that we've done some DNA replication. The genetic material, these sisters remain relaxed, okay? So they're not coiled yet, they're not condensed, they are in a relaxed state. As we proceed to the last step of interphase, the last phase, G2, essentially nothing has changed. You still have sister chromatids, and you still have them in a relaxed state. As we proceed into the mitotic phase here, with the first step of mitosis, that being prophase, there is a significant change. The chromosome is still duplicated. It is still composed of sister chromatids, one black and one red. However, the genetic material has condensed. It has begun that coiling up process, and it is now condensed inside that cell. Okay, so this compaction, this condensing of the genetic material facilitates the mobility of the sister chromatids. So if the DNA remained in a non-condensed or loose state, it would be susceptible to damage. Think of a giant ball of yarn, and if you pull on one facet of the ball of yarn, you may tangle another part of that ball of yarn. So it makes sense to condense and to be more compact in prophase, and that's the initial spot where you get this condensation. So as we proceed to metaphase, the second part of mitosis that I want you to know, not much has changed. The nucleus is completely gone here. However, the genetic material remains in the form of sister chromatids, and it remains compact. We arrive at a significant event in anaphase. That is essentially the separation of those two sister chromatids. So we can see that we have a separation event. And now each of these sister chromatids goes back to being called a chromosome. So these are two separate chromosomes. And basically it shows you now that we duplicate our genetic material in S, so that in anaphase, when we separate the genetic material, each new daughter cell will get a full complement of that genetic material. Okay, so our sister chromatids is split up. We now have chromosomes again, but the chromosomes remain compacted. That's all about to change, though, as we step over into telophase. As telophase occurs, our newly separated chromosomes begin to return to their relaxed state. So they decondense, essentially. They become relaxed in preparation for returning to interphase. The nucleus returns to protect them. The nucleus is very important. And then by the time we're at cytokinesis, we have now two daughter cells, each with that genetic material decondensed. Okay, so it's gone back to being relaxed. And essentially that's it. So I wanted to show you, to give you a better idea of where the genetic material is as far as it being duplicated or not, and if it's compacted or not compacted on the way to being separated. Okay, so I hope that gives you a better picture of the changes throughout the mitotic cell cycle, specifically for your genetic material. Let me know if you have any questions. I hope that helped, and we'll see you next time.